Today we're going to talk about the stages of protein synthesis.
I don't know anything about protein synthesis.
Neither do I.
Well, good thing I do.
Okay.
Hi, my name's Alexis, and I'm going to talk about translation initiation.
Hi, Alexis.
The process starts with a few key players that will allow for the future events of translation that will be discussed by my friends later. The 30S subunit, the 50S subunit, the mRNA, the initiation factors, and the pivotal AUG codon. They work to form the pre-initiation complex and make sure the machinery is properly aligned on the mRNA. The PIC forms through binding and aligning of the 30S ribosomal subunit to the mRNA.
With all the nucleotides in the mRNA, how can you be sure that 30S binds properly?
Well, you can be sure because the AUG codon specifically codes for FMET. Also, 8 to 12 base pairs upstream of the AUG codon is the Schindler-Garneau sequence of the mRNA that will base pair with this complement, the anti-Schindler-Garneau sequence, located at the 3' end of the 16S rRNA of the 30S subunit. This is critical for the positioning of the AUG in prokaryotes. The next important place to mention are the peptidyl site, or the P site, and the amino acyl site, or A site, of the 30S subunit. Simultaneously, IF3 will bind to the P site, and IF1 will bind to the A site. The IF3 acts to prevent the premature binding of the large 50S subunit and ensures fidelity by distinguishing FMET tRNA from the amino acyl tRNAs. IF1 works to stimulate the IF3 and IF2 activity. Next, the IF2 bound to the FMET tRNA FMET, and GTP will bind to the complex via pairing of the tRNA's anticodon UAC and the mRNA codon AUG at the P site. The complex is then bound by the 50S subunit. GTP is hydrolyzed and the initiation factors leave the complex.
Thanks, Alexis. That was great.
You're welcome.
Hey everyone, my name is Alitza and I'll be introducing the process of translation elongation.
Hey, Alitza.
As Alexis just described, once the initiation complex comes together and the appropriate tRNAs are charged with their respective amino acids, we're ready to go for elongation. This process requires activated tRNAs, 3 elongation factors, and GTP. Now that the tRNA charged with methionine has already recognized the AUG start codon on the mRNA and bound to the peptidyl site, the first step in prokaryotic translation elongation can begin. During this step, the second amino acyl tRNA recognizes its corresponding mRNA codon and brings the appropriate amino acid to the A site of the ribosome. In this case, we have valine.
How does the recognition occur?
The anticodon loop on the tRNA recognizes its complementary codon embedded within the mRNA. Remember, the anticodon on the tRNA is oriented anti-parallel to the codon in the mRNA. This tRNA anticodon forms 3 hydrogen bonds with the appropriate codon on the mRNA to help hold the two together.
What about the factors you mentioned? How are they involved?
The second amino acyl tRNA for valine binds specifically to elongation factor TU, which is bound to GTP on the initiation complex. Once the tRNA binds here, GTP becomes hydrolyzed. The elongation factor TU GDP complex is then released. And a new elongation factor TU GTP complex is regenerated for the next tRNA cycle. Now that the two tRNAs are side by side in the P and A sites, their amino acids must be linked through a peptide bond to start forming the polypeptide chain, which brings me over to Morgan.
I'm Morgan. And I'll tell you how translation continues to elongate the mRNA strand and how the process works.
Hi, Morgan.
So, what makes all these parts work together to make proteins?
Peptidyltransferase. It's an aminoacyl transferase, and its activity is carried out by the ribosome. It positions the 23S RNA in loops that allow it to interact and catalyze tRNAs. First, the P loop allows 3 nucleotides in the loop to bind to the peptidyl tRNA in the P site.
What about the A site?
The A loop binds to the aminoacyl tRNA in the A site and is another terminal loop. Then peptidyltransferase puts the A and P loops close to each other before moving the aminoacyl part of the aminoacyl tRNA to the ester bond of the peptidyl tRNA. A peptide bond is then formed as the aminoacyl tRNA amino acid nucleophilically attacks that ester bond.
What happens now?
Now the ribosome moves 1 codon down on the mRNA strand in the 3' direction. To do this, it requires EF-GTP and GTP hydrolysis. Then the dipeptidyl tRNA in the P site moves to the A site, leaving the deacylated tRNA in the P site free to move into the E site.
What happens to the A site?
The new peptidyl tRNA enters the freed up A site, and the old deacylated tRNA leaves the complex at the E site. E for exit, and the cycle repeats again.
How does the ribosome know when to stop translation?
The A site now contains the termination codon. There are 3 release factors involved. RF1 recognizes UAG and UAA. RF2 recognizes UGA and UAA. RF3's function isn't known. RF1 or RF2 release factor binds to the termination codon, promoting the 23S RNA to allow water to get into the active site.
What does water have to do with anything?
The oxygen in the water makes a nucleophilic attack on the ester bond, which attaches the polypeptide to the peptidyl tRNA. This attachment results in hydrolysis of the bond, and the finished polypeptide is released. Now the 70S subunits dissociate from one another, and translation has ended.
Hi, my name is Gabby, and I'm going to talk about regulation of protein synthesis.
Hey, Gabby. Wow, these students must work really hard all the time. When do they take a break?
They are on all the time. That would be a waste of energy and materials. They are turned on only when they are needed.
Whoa, that's smart. But how does it all work?
Well, let's look at the example of the trip operon. When the cell needs to synthesize more tryptophan, the trip operon gets turned on because those genes make the enzymes that are needed for tryptophan synthesis.
Wow, it sounds like there are a lot of levels to this stuff.
There sure are. The trip operon gets regulated in 2 different ways. The first is transcriptional repression. There's something called a repressor that binds to the operator region of the trip operon and stops transcription from occurring. In other words, it turns off the trip operon. When the trip repressor is not bound to the operator, tryptophan is continuously synthesized. The trip operon has a special repressor called an aporepressor.
What does that mean?
It means that the trip repressor cannot bind to the operator by itself. It needs help. Its helper is tryptophan. When there's plenty of tryptophan in the cell, it binds to the trip repressor and the whole complex binds to the promoter and stops transcription because it is not needed anymore. When there isn't enough tryptophan in the cell, it can bind to the repressor, and the repressor therefore can't bind to the operator. Transcription continues until the levels of tryptophan increase.
It's almost like there's one part of the cell that is telling the other part of the cell what it needs.
Exactly. It's called a feedback mechanism. The second way that the TRP operon is regulated is called attenuation, which means pausing. This level of regulation only occurs while transcription is going on. The 5' non-translated region contains 4 important sequences. Sequence 1 contains an open reading frame called the leader peptide that has 2 side-by-side TRP codons in it. These 2 codons are the key to this type of regulation. Sequences 2 and 3 are complementary, as well as sequences 3 and 4.
How can sequence 3 be complementary to 2 things at once? Can it bind both sequences at the same time?
It cannot, and that's the key. When there's plenty of tryptophan in the cell, the ribosome moves along the mRNA transcript, and when it comes to the double TRP codon in the leader peptide, there is enough tryptophan-charged tRNA to add tryptophan to the peptide sequence without the ribosome having to stop. Because the ribosome doesn't stop, there is not enough time for sequence 3 to pair with sequence 2. So it must pair with sequence 4. When this happens, a secondary structure forms that blocks RNA polymerase from continuing transcription because there is enough tryptophan in the cell already.
What happens if there's not enough tryptophan in the cell?
When the concentration of tryptophan in the cell is low, the ribosome has to stop at the double TRP codon as it moves through the leader peptide. This gives sequences 2 and 3 enough time to pair together. They form a different secondary structure that doesn't stop transcription. And both the RNA polymerase and ribosome can continue transcribing and translating the gene.
Wow, I had no idea that the operons could be so closely controlled.
Yep, and the tryptophan operon is not the only one. The operons for histidine, threonine, leucine, isoleucine valine, and phenylalanine are regulated by attenuation as well. Phenylalanine is one of those that is also regulated by a repressor also.
Thanks, Gabby. Genes are so cool.
Whatever happened to predictability? The milkman, the paper boy, evening TV. Did I get delivered here? Somebody tell me, please. This whole world's confusing me. Clouds as mean as you've ever seen. Ain't a bird who knows you're true. Then a little voice inside you whispers,